Welcome to our channel Mootechie. This is where we dive deep into scientific concepts with clarity and curiosity. Theories of biogenesis and abiogenesis. These are the two major frameworks scientists use to explain how life began and how it continues. Exploring the origins of life, from prebiotic chemistry to cellular complexity. We'll trace the journey from simple chemicals to the first living cells, highlighting key ideas and experiments along the way. Origin theories. We'll compare different explanations for life's beginnings. Key experiments. We'll look at landmark studies that shaped our understanding. Microbiology impact. And we'll see how these theories influence modern science, especially in fields like microbiology. Now, let's begin with a foundational question: the origin of life. Introduction: Origin of life. Life emerged from prebiotic chemistry rather than spontaneous generation. This means the first life didn't just appear from nothing, it formed through gradual chemical processes on the early Earth. Early Earth 4.0 to 3.5 Ga had a reducing atmosphere rich in simple gases. Billions of years ago, the air was very different, filled with gases like methane and ammonia, not oxygen, which created a unique chemical environment. Key ingredients: water, energy, lightning/UV, and organic monomers. These basic components, along with energy sources like lightning or UV light, provided the raw materials and power for chemical reactions. Candidate settings: hydrothermal vents, tidal pools, and glacial ice. Scientists think life could have started in these diverse environments, each offering unique conditions for chemistry to become biology. Central question: how did chemistry organize into biology? This is the core puzzle. How did non-living molecules cross the threshold to become something alive, capable of growth and reproduction? In this slide, we examine a major scientific hypothesis for that very question. Scientific hypothesis: theory of abiogenesis. This is the idea that life can arise from non-living matter through entirely natural processes. Life arises from non-living matter via natural processes. It proposes that under the right conditions, simple chemicals can spontaneously give rise to living systems. Evolutionary stages: monomers to polymers to protocells. The process likely happened in steps: small molecules linked to form chains, which then assembled into bubble-like structures that resemble simple cells. Central pathways: RNA world, metabolism first, and lipid world. Scientists debate which came first: genetic molecules like RNA, self-sustaining chemical reactions, or cell membranes. Key drivers: energy gradients, chemical catalysis, and self-assembly. Factors like heat differences, mineral surfaces, and the natural tendency of molecules to organize themselves pushed this chemical evolution forward. Core prediction: testable chemistry is possible under early Earth conditions. The theory is scientific because it makes predictions we can test in the lab, trying to recreate the steps that might have happened long ago. Let's look at some of the key experiments that support the idea of abiogenesis. Key evidence: experiments supporting abiogenesis. 1953 Miller-Urey experiment. Simulated early atmosphere H2O CH4NH3H2 with electric sparks. Result: produced amino acids glycine-alanine from inorganic precursors. This groundbreaking experiment showed that the basic building blocks of life could form naturally from Earth's early atmosphere and lightning. 1961 Juan Oro's synthesis demonstrated that adenine, a key DNA-RNA base, could be synthesized from hydrogen cyanide (HCN) and ammonia. This proved that even the complex information-carrying molecules of life have plausible non-biological origins. 1960s Fox's proteinoids. Heated amino acids polymerized into thermal proteins. These formed microspheres exhibiting basic cell-like properties. By simply heating amino acids, researchers created structures that could divide and maintain an internal environment mimicking early cells. 2000s, SOSTART and protocells. Research on fatty acid vesicles and RNA templates showing how the primitive cells could grow, divide, and evolve. Modern work shows how the first cell-like compartments could encapsulate genetic material and undergo a simple form of evolution. Ongoing, mineral and vent catalysis. Role of clays and iron-sulfur minerals in concentrating monomers and catalyzing polymerization near hydrothermal vents. Scientists now study how underwater hot springs and certain rocks could have acted as natural laboratories, speeding up the assembly of life's molecules. The Miller-Urey apparatus. This image shows the classic experimental setup, a flask representing the ocean and an atmosphere chamber where sparks simulated lightning, driving the formation of organic compounds. Now, let's turn to the other major theory which governs life as we see it today. Theory of biogenesis. This is the principle that life comes only from existing life. Life arises only from pre-existing living organisms' reproduction. This is a fundamental law of biology we observe every day. Cells divide, animals reproduce, but new life doesn't spontaneously generate from dirt or air. Replaced ancient notions of spontaneous generation like maggots from meat. For centuries, people believed life could spring from non-living things, but biogenesis replaced that old idea with evidence. Driven by mechanisms like cell division, reproduction, and heredity. Life continues through specific observable processes that pass genetic information from one generation to the next. Implies unbroken lineage continuity tracing back to ancestors. Every living thing today is connected by a continuous chain of reproduction stretching back to the very first life form. Foundation for germ theory and modern aseptic techniques. Understanding that microbes come from other microbes, not from the air itself, was crucial for developing medicine and sterile procedures. Diagrams. This slide shows diagrams of cell division, specifically mitosis. On the left, we see pro-metaphase with structures like the spindle pole, kinetochore microtubules, and chromosomes aligning. On the right, telophase is shown, where the nuclear envelope reforms around the separated chromosomes. These diagrams visualize the precise replicable process of biogenesis at the cellular level. This slide walks us through the historical experiments that definitively disproved spontaneous generation and proved biogenesis. Experiments supporting biogenesis. 1668, Francisco Redi's jars showed that maggots on meat came from flies, not the meat itself. Sealed or gauze-covered jars remained free of maggots. Redi's simple but clever experiment demonstrated that living larvae required parent flies to lay eggs, challenging the idea that rotting meat generated life. 1765, Lazzaro Spallanzani. Boiled broth in sealed flasks showed no growth. Critics argued sealing excluded the vital force, air. By sterilizing broth and sealing it, Spallanzani prevented microbial growth. Critics' objections about air actually paved the way for Pasteur's even more conclusive design. Louis Pasteur's swan necks used curved flasks that allowed air in but trapped dust. Broth remained sterile, proving microbes come from air. Pasteur's ingenious swan neck flasks were a masterstroke. They allowed air, the supposed vital force, to enter, but trapped dust-borne microbes, proving that life, microbes, comes from outside, not from spontaneous generation in the broth. 1870s, John Tyndall explained inconsistencies by discovering heat-resistant endospores, developed fractional sterilization, Tyndallization. Tyndall solved a puzzle why sometimes broth spoiled even after boiling. He discovered bacterial spores that could survive heat, leading to more effective sterilization methods. Conclusion: Omne vivum ex vivo. Established the law of biogenesis, life arises only from pre-existing life, refuting spontaneous generation. This Latin phrase, meaning all life from life, became the definitive statement of biogenesis, a cornerstone of modern biology. Pasteur's swan neck experiment. The image illustrates the setup. A flask with a long curved neck is boiled to sterilize the broth inside. Even with air contact, microbes get trapped in the neck's bend, keeping the broth sterile. Only when the neck is broken do microbes fall in and contaminate it, visually proving that microbes come from outside, not from spontaneous generation in the broth.
Now that we've seen both theories in detail, let's place them side by side for a clear comparison. Comparative analysis. Abiogenesis versus biogenesis. Abiogenesis. Source. Life originates from non-living chemical matter through natural evolution. This theory addresses the ultimate one-time origin event on the ancient Earth. Timescale. Occurred on the primordial Earth around 4 GA under reducing atmospheric conditions. It's an event confined to a specific, distant period in our planet's history. Evidence. Supported by lab simulations, Urey, and models of chemical evolution. The evidence is indirect but compelling, coming from chemistry experiments and theoretical models. Scope. Explains the origin of the very first life forms from simple molecules. Its job is to answer the profound question of how life initially emerged from non-life. Biogenesis. Source. Life arises only from pre-existing living organisms, reproduction. This is the rule that governs all of biology we observe in the present day. Timescale. Occurs continuously today, the standard mode of life propagation. It's not a one-time event, but the ongoing process driving the diversity of life. Evidence. Proven by direct observation and experiments by Pasteur, disproving spontaneous generation. The evidence is direct and observable. We see cells dividing and organisms reproducing every day. Scope. Explains the continuity and diversity of life once it exists. It describes how life perpetuates itself and changes over time through inheritance and reproduction. Understanding biogenesis isn't just historical. It has immense practical importance in modern microbiology. Importance of biogenesis. Aseptic technique. Strict protocols to maintain sterility, preventing the introduction of unwanted microbes into experiments. Because we know microbes come from other microbes, we can design procedures to keep our work areas and tools completely free of contamination. Germ theory. Understanding that specific diseases are caused by specific microorganisms, not spontaneous generation. The rejection of spontaneous generation was essential for realizing that diseases are caused by living germs that spread, leading to modern medicine. Pure cultures. Methods to isolate single strains of bacteria for study, essential for Koch's postulates. Biogenesis allows us to grow and study one type of microbe at a time, which is fundamental for identifying the causes of diseases. Contamination control. Identifying sources of biosecurity risks. If life appeared spontaneously, controlling contamination would be impossible. In labs, hospitals, and industries, we can trace contamination to a source and eliminate it, precisely because life doesn't spontaneously generate in our sterilized environments. Sterilization protocols. Basis for autoclaving and disinfection. We know heat or chemicals kill existing life, leaving the medium sterile indefinitely. Our entire approach to sterilization relies on biogenesis. We know that if we kill all existing microbes, no new ones will spontaneously appear, so instruments stay sterile. As we reach the end of our presentation, let's conclude our exploration. Thank you. We hope you enjoyed exploring origins of life with us. More science on Mootechie. We have plenty more topics where this came from, diving into other fascinating corners of science. Subscribe now. If you found this insightful, consider subscribing to stay updated on our latest content. Questions? Drop a comment below. We encourage your curiosity and are here to help clarify any questions you might have.